Բարեցես, բարի լույս, բարի գուն, բարի այսօր, գախումնի որտեղ կգտնվիք։ Այս panel-ը պիտի ինենք անգլերենով, այնպես որ հետո անգլերենով պիտի սկսեմ ես հիմա։ Again, thank you for joining us. This is the ARPA Institute panel discussion series. I'm Hagop Panosyan, the president of ARPA Institute, and I would like to say a few words about our activities in Armenia. And then I will introduce the moderator today, Dr. Sargis Sedrakyan, who will then introduce the panelists, and then we will start the panel discussion. ARPA Institute has been working in Armenia for the past 33 years, and we have achieved various projects. Our main goal is to advance science, technology, healthcare, and other areas. ARPA is a organization of experts from various fields, and our objective is, as the name stands, analysis, research, and planning for Armenia. That's what we do. We analyze the situation, we talk with people, find out what is available and who is doing what, and we try to analyze their activities and our capabilities, and then accordingly plan, decide whether we can achieve what is needed, and then start working. Our current projects are on various areas. One of the most important one is we have established a new advanced circuit design and modeling center in the Polytechnic University. This is now the second year. The first year was essentially a starting year, the last year, but this year they have already 120 students participating in courses related to circuit design. And the circuit design courses that are being taught is provided by Cadence Systems, which is the software that is internationally recognized, and essentially all the large companies of circuit design use it. And Cisco Systems is also, of course, helping very much, especially the manager in Armenia, uh, Fahe Yeryazaryan, is helping both teaching the course and also organizing the activities. And the second one is we have built, of course, the first clean room, class 1000 clean room in Armenia in the National Polytechnic, National Alekhanian Lab. And we also work in other teams with the Alekhanian Lab. One of them is the radio frequency photomultiplier tube, which is essentially an instrument that is able to detect laser pulses down to a picosecond accuracy, which is we believe, a very important achievement. And and then we also have the detection lab, the nuclear detection lab, which is another very advanced lab that we support. We also work with the universities, obviously, and also other institutes of the academy. We have worked with the Molecular Biology Institute, the Physical Chemistry Institute, the Chemical Physics Institute, and other institutes. And that's what we do in Armenia. We also have a annual invention competition for young scientists, and we have this year received 12 inventions, and they are now being evaluated. The first phase is already over. The invention competition is two phases. The first phase is we ask inventors to send an extended abstract with graphs and pictures so that the expert will evaluate their main idea and the concept. And if it is found worthy of further evaluations, we will select those that are considered better than the others, and we will ask for full proposal. And then the second phase will involve the evaluation of the full proposal, and the experts will determine the best of all, and the best team will receive $10,000 award. And of course, we also organize this, panel discussions and sometimes presentations related to Armenia or Armenians in general. Genomics, today's topic is genomics and AI, artificial intelligence applications on projects in Armenia. And we are happy to have the esteemed panelists today, Kristine Margaryan, Lilit Nersisyan, and Hovakim Zakaryan. The moderator is Dr. Sargis Sedrakyan. Sargis has been with the ARPA Institute serves now as the treasurer of ARPA Institute. He's an expert in DNA analyses related to cancer and other areas, and he's also worked with Armenia. And now, Sarkis, the floor is yours.
Thank you, Dr. Panelists. Good morning, everyone, and welcome to the ARPA Institute's panel discussion on genomics and AI applications on projects in Armenia. As Dr. Panelists already mentioned, we will be discussing around genomics and AI applications and the future of this discipline in Armenia. Thank you for all of you joining today to this panel, whether you're tuning in from Armenia, from locally here in the United States or Europe or elsewhere around the world. And I'm looking forward to an engaging and interesting discussion throughout the next hour or so. So Armenia is at a critical, turning point where genomics, computational biology, AI are no longer emerging concepts, but rather they are becoming essential tools that drive education, namely STEM education, research and innovation in healthcare, agriculture, and medicine in the 21st century. And today we're fortunate to have an outstanding group of panelists whose combined work spans both industry and academia, and who are shaping that transformation on the ground every day in Armenia. So throughout today's discussion, we are going to highlight the powerful intersection of genomics. We'll touch upon its role in agriculture, microbiome science, biotechnology, and artificial intelligence, as well as their essential applications in Armenia. So particularly, we will explore advances in plant genomics and the National Grapevine Collection, innovations in computational biology, and the bioinformatics capacity, which is very important, to provide the capability to perform all of this analysis, and emerging efforts in AI-driven drug discovery and antiviral development. So taken together, these efforts offer a glimpse of Armenia's scientific future, a framework poised to shape and accelerate the country's technological growth in the decades to come. So without further ado, I am honored again to introduce our distinguished panelists. Dr. Kristine Margaryan, she's the head of the Plant Genomics Research Group at the Institute of Molecular Biology, a senior researcher at the Laboratory of General and Molecular Genomics at Yerevan State Medical University, and the manager of the Armenian VITUS database. I hope I'm pronouncing that correctly. Next, we have Dr. Lilit Nersisyan. She's a computational biologist and the founding director of the Armenian Bioinformatics Institute. And lastly, Dr. Hovakim Zakaryan, who is a leading virology expert. He's the head of the antiviral research group at the Institute of Molecular Biology and also the co-founder and CEO of De Novo Sciences. With that, let us begin our conversation. But before we do so, let's take a moment to clarify for our audience what is genomics. And so I would like to ask our panel to give a very brief overview of what genomics is. It's a big word. It encompasses different areas. So if you could break this down for us, and then each of you will take a few minutes to describe and define the projects that you have, your own projects involving encompassing genomics and artificial intelligence, and what are some of the recent discoveries and interesting areas that you are exploring. And before I uh, pass you the microphone, let me address the audience. So throughout these discussion, please, if you have any questions, you can use the chat option. You can enter your questions and we'll get to them towards the end. Or at the end, we'll open the floor if you want to turn on the microphone and you can ask your question directly to the panelists. So let's get started. Maybe if Lilit can give us a brief background and some definitions of what genomics is and how is it, for example, different from um, genetics, genomics and genetics. What is the difference?
Yeah, thank you, Sargis, for the introduction and to our Institute again for organizing this and for the guests to for taking the time to be here. Basically, the suffix omics means the collection of. So if the field of genetics has evolved from started from studies of single genes when people try to understand if this gene, for example, is responsible for the color of this flower or for the shape of this pea, etc. Then, of course, science understood that genes in isolation, they are not answer to all the questions, and that the genes and their products, they work in combination when they interact with each other, etc. So the combination of all of the genetic factors in a cell is responsible for the cell's physiology, for the cell's behavior, and for the organism's phenotype. And basically, if you study all of the genes in combination all together, then you name this field genomics. And right now, people not only refer to the study of DNA sequences as genomics, but they also tend to refer to all the layers. Basically, DNA encodes RNA, and if you study all the RNA in a cell, then you get the transcripts. So basically, everything that is read from the DNA and turned into RNAs, and basically this process is transcription, hence the word transcriptomics. So if you study all of the RNA, that's transcriptomics. If you study all of the proteins in the cell that are produced eventually, then this is proteomics, etc. And sometimes people say genomics is the study of all the genetic material that we have in our bodies, but sometimes they also refer to other modalities of data. So basically, genomics is a very general term to describe all the code that we have in our body that is used to form our phenotype, basically.
And just to follow up on that, why is it important to study all the genes as versus individual specific genes that define, the pea color, for example, that you defined? Why is it important to study the
Um,
entire genome?
yeah, so maybe an example of like how our diseases are formed. a part of the diseases that we study, they are monogenic. So if you have a mutation in one single gene, like for example, the familiar Mediterranean fever or Yerevanyan disease, then you will have the disease. However, there are also complex disorders where a single mutation in one gene is not the answer to everything. You maybe need to have the combination of like several mutations. And on top of that, the interaction with the environmental factors in order for the disease to form. Sometimes one gene is not enough to form a phenotype. Sometimes you need to have all of the things. And basically, we can get the equivalent of the state of science in Armenia. If you have one scientist cannot rescue the situation. You need to have like a critical mass of a lot of different players and not only scientists, all the factors that form the ecosystem in order for the thing to move forward. And the same thing happens inside the cell. So you need to have a lot of factors that together regulate the whole ecosystem. And unless you look at the big picture from above, you maybe will have this tunnel vision and you won't be able to understand a lot of mechanisms.
Thank you, Lilit. Thank you for providing that overview. Obviously, it's very complex and requires a lot of expertise to be able to do that. So let's take a few minutes now for each of our panelists to give an overview of their respective projects in their field of study, so we can set the tone for today's discussion. We can have Lilit go first since she started.
Yeah, sure. So basically, I study bioinformatics, and this is branch at the intersection of computer science, maths, statistics, and genomics, basically. And what we do, we turn data into knowledge. So the data in our case is all the genomics in its wide term. So basically DNA sequences, RNA sequences, epigenomics, etc. And we given the data from different scenarios, we try to come up with biological knowledge. Examples of that are, for example, cancer genomics, where one of our teams is trying to understand the genomic basis of heterogeneity, of how the patients develop the disease and how they respond to different therapies, and what is the reason that some patients do respond and others resist. We also have microbiome research team, and here we study all the diversity of microbes in our organisms, in the gut, in the oral cavity, etc., and how the composition of this microbial ecosystem changes during health and disease development. We also look into telomeres, the protective caps of our DNA that are related to aging and cancers. And we also look into non-invasive diagnostics. Basically, we try to identify biomarkers of disease in freely circulating DNA pieces in our bloodstream that could indicate about possible disease. And all of this we do, of course, using computers to analyze data.
Excellent, thank you. Kristine, I know your work focuses on characterizing and preserving Armenia's native grape wine varieties. Can you please tell us about that and your recent book also on grapes of Armenia, which I haven't had a chance to read, but if you could tell us more about it. I think it contains some incredibly important information, if I'm not mistaken, characterizing around 182 different varieties of grapes in Armenia. Please take us through it.
Thank you, Sargis. I would like to say hello to everyone for joining to our discussion. So I'm Kristine Margaryan, as Sargis represent me already. So I'm head of the research group of plant genomics. And for now, I guess we are the only group in Armenia working with wine genetics and the genomics, and not only, because recently we started a project also related to the wine breeding. And again, we are the only group working with wine breeding as well in Armenia. So we are working on these topics. We call it like 3C concept. It's collect, characterize, and conserve. Armenian, first of all, native, indigenous, neglected grapevine varieties. And almost 15 years we are following this concept. And for now, we have genotyped or sequenced more than 3,000 different accessions, almost 200. For almost 200, we have a whole genome sequences data. And what is very important and one of the main outcome for us, it's trying to document all the precisely done genetic and phenotyping passport, not only in Armenia with this database, but also to the international to confirm the origin of Armenian grapevines, because that becomes quite strong dilemma, especially with Azerbaijan. And each time when we are recovering new or neglected varieties, we documented it also, as I told, in European with these databases where the origin noted Armenia. So for now, we have registered more than 400 different native Armenian grapevine varieties in with this international variety catalog. And this year, I hope we will have plus 50 new grapevine varieties. And working almost 15 years, there were gathered a lot of information, huge information, and a huge data set. And with my colleagues, Erika Maul, she was almost 30 years, the only responsible person for this international variety catalog and one of the well-known ampelographers worldwide. And we decided that we need to publish our data, not only like article, but finally, for the first time, to publish and to create really important data. It's not a book, it's like a database for everyone who is working in wine and wine sector. As you told, yes, there are involved 181 Armenian grapevine varieties, from which 117 are autochthonous neglected varieties. We focus mainly on the neglected because we found sometimes and quite often only 1-2 plants preserved in abandoned garden in very old vineyards. So we decided that this is critically important to collect and to conserve first of all them, rather, let's say, Areni or Boskehat, because there are quite you can find them they're everywhere. So and 56 are bread cultivars, also less known but with great wine potential, let's say. And for the first time, we included also some wild grapes, because the recent 5 years, one of the project of our group working. The idea is to work and to uncover breeding potential of Armenian wild grapes, because we have described and discovered new R markers which make them absolutely resistant against different diseases like powdery and downy mildew. This was really unexpected some results, because actually we were known that these R markers, they always comes from American or Asian grapes. But it seems that we updated, as once told me our colleagues from Europe. She told you, thanks to this diversity, we are updating our knowledge about grapevines. So there is really hidden potential, and we are working to uncover it. And why this book is really very important, because for the first time, we apply like symbiotic approach. We put all together not only genetic data, I mean not only genetic passport by 25 SSR markers, but also quite precisely done ampelographic characterization for each variety. And what is very impressive, that everyone look on the book told that it's pictures. images. So I will transfer each plant, each variety from the vineyard to the special photo studio, and we did a special photo session for each variety. So according to the European standards, according to the OIV standards, we took a picture for all the bunches, separated berries, the leaves, the young shoot, etc., and all the characteristics for each organ down. And right now it's on the publishing process. I hope we will do presentation before New Year or just after New Year. It depends how fast they will do the publishing process. So, but what is important also that the title of our meeting today is Genomics and AI. So in terms of grapevines, about 5 years we work quite hardly with morphometry as well, and I have an idea why I need to sit and to spend a lot of time to measure some characteristics. I need to collect data, and I just asked my colleague working in AI company, and with one of them we have already signed an agreement to work together, and the guy created fantastic tool for us. And just for example, to understand how it will accelerate the process of my group, for each year, we were able to characterize by morphometrical descriptors almost 20 varieties. Now, by this tool, we are able to characterize, I don't know, he told about 10,000 images during 15-20 minutes. You just need to upload your images, and the AI tool will characterize and will group all the images according to the descriptor you train him to recognize. So this is our first chance to work with companies working in AI, and I hope we will continue because there are a lot of other ideas how AI can accelerate the process in wine genetics and the genomics as well. So, and what is the next? So I'm very happy to say that almost after 30 years of break, we started to work also on the breeding because, you know, after collapse period of Soviet era, unfortunately, there were no one who continued to work in wine breeding. But during Soviet era, the wine selection and the breeding was quite actively done, and almost 450 different varieties have been created for that period. And unfortunately, there were like a generation gap. We were not the generation who met these old scientists for knowledge share. So we have no knowledge how to do the process. And thanks to our cooperation with a German institute, and with them we have 15 years of close cooperation. That's well-known Institute of Grapevine Breeding, So this year we already will have the first F1 generation, where we try to transfer these R genes from wild grapes into Armenia and the Bosquehat, trying to meet also some challenges related to climate change and the biotic stresses. So, and in spring already we will start a new sequencing a process to see how successful was the crossing and if the R genes we were able to transfer from wild into cultivated grapes. This is, I don't know, really a priority task, not only in Armenia, but all over the world, because now everywhere it's tendency to move to organic viticulture and the organic winemaking to start to reduce the application of different pesticides. So, and we did the first like a step to go forward to these challenges, and we are continue to to collect and to discover and to rediscover neglected varieties. And for now, we have registered 176 varieties which were never described, never documented. So we found them in abandoned garden in Sunik, in Tavush, in Wildstor, few of them also in Ararat Valley. But the big surprise for us was Sunik region, because in all the stampelographic books, you will never find any information about the great potential of Sunik grapevines, except like Spitak Arabic. This is the only known grapevine varieties. But only this year, I have collected more than 100, also wild grapes. And for this year, we have collected plus 300 new, never described grapevine accessions. And right now, I am on the process of identification, and at least 150 to 200 we expected to have a new genotypes. And so this is really a treasure to have a chance to work with this huge biodiversity. But what is very important to convert this data to applied science also, because all the projects, the main outcome of our projects need to be applied also in practice. So, and I hope once in Armenia also, the sector will recognize the importance of science we are doing in our lab, and they will try to use this data in practice as well. So, but there is some movement recent years, because I have a lot of requested from different winemakers and the farmers. When they found some new unknown ones, they call me. We found you can collect, you can analyze, you can recognize, which wasn't the case, let's say, 10 years ago. So I guess there is some positive response already from the sector, and we are waiting some positive response also from governmental bodies as well.
Wonderful. Thank you. Thank you, Kristina, for that interesting insight and overview of your studies. And given the importance of wine production in Armenia, I think whatever you're doing is incredibly important. And hopefully, the government and the private sector can actively participate in developing the knowledge and information generated through these projects to something tangible and importance for Armenia's economic development. We'll touch more upon this. We'll come back and have a lot of questions, obviously, but I would like to give a chance to Hovakim to take us through his endeavors in virology and how he uses artificial intelligence in discovering antiviral, targets.
Thank you. So, good evening and good morning. Thank you for having me in this panel. It's a great pleasure to join you. So, I started my journey in virology like around 15 years ago, when I started to do my PhD in the laboratory of Zakaryan in Institute of Molecular Biology. And then since 2016, I run my own group focusing on discovery of small molecules with antiviral properties. In the beginning, the only virus that we had in the lab was African swine fever virus, which is not dangerous for humans, but it's really very big headache for pig industry, pork industry, because of lack of vaccines and antiviral drugs against that virus. But since that time, step by step, we added the viruses in our biobank, and right now we have more than 17 different human and animal viruses in our lab, which is, I think, the biggest one in the whole region. We have different viruses from different viral families, DNA and RNA viruses. So when I started the laboratory, it was very clear for me that there is no funding allowing me to screen thousands or 100,000 of compounds as my partners or colleagues usually do in EU or in the United States. So, and on the other hand, even if I have some funding, still purchasing some compounds is very, very long and bureaucratic process, unfortunately, until now. And you have to wait months until you get some compounds for making the screenings against the viruses. And that demand or that need or that reality, like it triggered me, it stimulated me to look over and try to find new approaches allowing us to decrease the number of compounds, decrease the number of chemical compounds that we want to screen to find something. And this is how I went to computational biology in general. We started to explore whether we can use simple techniques like virtual screenings, allowing us to screen 100,000 of compounds virtually in the virtual environment, and then just come up with a list of like 20 or 30 compounds that we can purchase and screen using our viral and cell models. But then we also realized that even the best virtual screening approaches are not accurate at all. And when you are performing virtual screenings and then compare the results of virtual screenings with existing data, you see no correlations between the performance of your even the best virtual screening programs with the existing data. And this is like a pain point for us. And then I met with my two co-founders, Vartan and Meher, a few years ago. They are brilliant mathematicians and data scientists. And we started to discuss whether we can use AI to find small molecules. And of course, the initial goal was to find some small molecules that we can also test in the lab, because we had that environment and we had all facilities that allowing us to test small molecules. So by that time, it was like the beginning of 2020, before the hype in generative AI, before all ChatGPTs, Gemini, and something like that. We realized that generative AIs are quite limited. Even more classical AI approaches, machine learning approaches, are very much limited in terms of using them in pharma industry because of lack of data. You don't have this luxury of data as you want to have, because unfortunately there are no like million and million or billion and billion small molecules tested against the target that you can use to train to feed your models and then get some relevant models that you can use to design or screen small molecules. And that was the reason why we started to explore whether there are other AI approaches overcoming these data scarcity or data limitations. And we came to reinforcement learning, which was quite new approach in AI. If you go back to 2016, 2017, the group called DeepMind, which was not familiar at that time, but it's right now, they are superstars with their Demis Hassabis that just got Nobel Prize last year. But they published a paper in Nature demonstrating that reinforcement learning can learn to play Go and chess in the virtual environment, in the virtual simulation, by playing against itself thousand and thousand times. And as a result of that virtual simulations, it learns the basic rules, it learns how to play Go or chess, and at the end, it won the real game against Go champion. So we ask ourselves whether we can do the similar thing, but instead of playing Go or chess, let's play chemistry. Let's play to design small molecules with superior drug-like properties against the targets of interest using the similar approach. And this is when and where Vartan and Meher came on the stage and started to develop the algorithm from the first principles. It's not like just adaptation of existing reinforcement learning, but it's development from the first principles, a very new approach, very new algorithms that allowing us now to design small molecules, like just within 2-3 days in the virtual simulations. And it allows us to generate thousands of small molecules with the best drug-like properties, with the best ADMET properties, and the best affinity against the target of interest. And they are highly novel. You can synthesize them still, because in some cases, if the molecules are highly novel, you cannot go and synthesize them because of the complexity and because of many, many other issues. But our molecules, most of them, you can synthesize, and we synthesize a lot of molecules and tested more than 300 molecules over the time against different targets. And we really achieved very good performance because if you compare with the virtual screening campaigns, usually the industry average there is around 10%, which means that you need to screen, let's say, 100 molecules to find 10 molecules with some activity against the target. So this is average standard. In our case, the hit discovery rate is about 30%, which is like 3 times higher than other computational approaches, which makes us completely different from many, many other companies and approaches. And we use our technique to find some small molecules against viruses as well. So we tested the first synthesized molecules against influenza A and B viruses, and we performed many, many experiments, not only on cell models, but also in animals, demonstrating that compounds are viable and they are very potent even in the animal models. And right now we are in a point where we are collaborating, partnering with many different companies from different countries, from Singapore to UK, from Qatar to India. So, and we are delivering our technologies to other companies that are interested to use them. And also we are partnering with different organizations in co-development or co-discovery programs. And I will be happy later to discuss also or to tell you about the results. But in general, if I summarize like my journey until to this point, probably the most important thing that demand or need, it stimulated me to find new approaches. And I came to virtual screening and then switched to machine learning thanks to my co-founders and colleagues. And now we successfully use these new approaches developed by our team to find different therapeutic candidates, including antiviral agents.
Sargis, you're muted. Sargis, you're muted.
Oh, I'm sorry about that. Yeah, thank you, Hovakim, for that interesting introduction. I personally have been following your research and have been incredibly impressed by the progress you've made. And thanks for that overview. I have a quick follow-up question. You mentioned about reinforcement learning. Is this something that applies to identifying molecules to target viruses, or this can be generalized to, for other forms of drug discovery?
Yeah, the algorithms that we have developed, they are absolutely target agnostic, so we can apply them against any target of interest. And the only input data that you need to provide to the algorithm is the structure of your target. So even the structure could be not only protein, but RNA as well. So it's also possible. And the second input data, which you can provide to the algorithm, is the starting point from which you want to grow your molecules directly in the binding pocket of your target. That could be a simple, like single carbon atom. It can be some heat candidate that you already identified through other methods. So, and depending on the task, you can try to find new hit candidates or even optimize existing molecules, trying to improve some important properties like ADMET or drug-like properties or whatever you want to do. So, but it's target agnostic. I mean, we didn't develop it for viruses or viral targets. We tested the first molecules against the viral targets because of my lab facility that we have. But in general, we develop it for any target of interest.
That's incredible. Thank you. I see that we have questions coming in. Actually, it says it's hard to hear Christina. So if you could probably speak up, Christina, when it's your turn. I guess it's I could hear it okay, but perhaps it's difficult to follow. We have questions coming in. We'll get to those. So please, I encourage you all to enter your questions, or like I said earlier, you can also unmute yourself when we get to that point and ask your questions directly. What I would like to do now is zoom back a little bit and ask a more general question to the panel, and you can all respond to this. And how would you assess Armenia's current position in the global AI and genomics landscape? In other words, what does Armenia have in terms of infrastructure, talent, right, and market potential? Where, in other words, where are we in this spectrum? Are we at the beginning? Have we developed somewhat? Are we can we compete, or do we need a long way to go? What is your view? Who wants to tackle it first?
Okay, I will try to answer on the AI part. So if you look at the number of publications each year that we have on data science and AI, so we are like countries like from Africa or some other underdeveloped regions because the number is very, very low. So this is one of the metrics how you can measure the current situation on data science in general, because you may say that there are hundreds of startups, companies doing some interesting deep tech work, but still, if we are speaking about science in general, one of the measures is publications. So if you look how many papers each year have published on data science and AI, so nothing optimistic is there. So and even if you compare us with our neighboring countries like Georgia or Azerbaijan, so the similar situation we have on data scientists in general. I mean, this is something that really for us like pain point, because we are constantly looking for people that we can hire and work with them, especially on the machine learning part, data science part. So unfortunately, I mean, if you look at young researchers, young scientists, usually when after the second or third year of university studies, usually they are hired by some companies that are not doing some deep tech or science-oriented work. They are quite good companies. They are probably future unicorns, but they are not scientific companies at all. So they are hiring these good candidates, these let's say beautiful minds, and then they are not working in science at all. So this is what we have. So we are trying to find people that we can hire, and like month by month, it is more and more difficult to find some good candidates. So recently we hired some new team member from Russia, from Moscow. So she relocated to Armenia. Before that, we hired another scientist from Iran, from Tehran, and he worked with us for a long time. And the reason why we are looking around outside of Armenia, as I mentioned, because inside in Armenia we don't have good candidates that we can hire. And one reason is because of some companies, they are doing very aggressive hiring process from the second, third year of university studies, and they're hiring the best candidates and offering high salaries. But unfortunately, these companies are not science-oriented companies. And the second reason is that human pipeline is very, very limited here in Armenia. So unfortunately, there are some significant fields like chemoinformatics. We don't have any chemoinformatic studies or chemoinformatic classes in universities until now. Thanks to Lilit, Arsen, and some other scientists that are advancing bioinformatics, and we have now bioinformatics in general. But the situation is not the same for chemoinformatics, which is a very important field, especially if we are speaking about computational drug discovery. So more or less better situation with data science in terms of talent pipeline. But as I mentioned, the best talents are hired by some companies, or they go out from Armenia and continue their studies abroad, and they just don't return back. And this is also another reality that we have. So I hope I answered to your question.
Yeah, thank you, Hovakim. Do you have anything to add, Lilit or Kristine, in this regard?
In my case, what I can add, the only positive things in the field I am working, that's existence of huge biodiversity and the totally lack of experienced specialists, especially when you would like to do some comprehensive project with different layers, etc. And besides geneticist, you need a viticulturist, you need an agronomist, you need someone who is working in plant protection. Unfortunately, there is no one. There are only few quite experienced viticulturists which are working in different private companies. They have very limited time. They have no interest to join scientific projects. And we really have a huge problem with human resources. So in my group, I have two very motivated PhD students and two researchers, and that's it. So that's very hard also to involve young generation to go to work with you, because they don't want to go through the vineyards to collect material when it's raining, when it's sunny, etc. So as I told, the only positive things is the existence of huge biodiversity, and that's it. It's a really huge problem to find in that field very well educated with good experienced people, and then to involve them in scientific projects. So
as you were
Yeah,
answering.
maybe I can add a bit. So basically, intersection of AI and genomics. I think we can mean like understand different things when we say that. One of the classical things is use of generative AI, and particularly different types of foundational models that people right now try to apply to different fields of genomics. For example, they applied to single cell data, and they try to understand, for example, gene expression, understand how different DNA sequences or DNA elements regulate the expression of different genes, etc. And a lot of scientists have actually started from this, because single cell data is basically genomic data extracted from every single cell in a given tissue. So you can, from a single snapshot, have like hundreds of thousands of different data points. And this is a number that at least is acceptable for AI. If you take the rest of the biological applications, usually you have 6 mice, sometimes you have 20, sometimes you have 100 patients, depending on what you're studying. But the amount of data that is generated in the field of genomics is usually not enough for this kind of foundational models to be trained. Of course, the field is moving in that direction, and sequencing is becoming cheaper and cheaper, and rich entities, they are able to generate enough amounts of data to also develop foundational models in different fields. For example, there's attempts to make foundational models on DNA sequences, on microbiome. For microbiome, it's too early yet, but there are attempts that are not working, but at least people are working in that direction. And the other thing is basically agentic AI, where you don't develop foundational models, but you try to use AI in order to automate pipelines in order to make an AI scientist or to help to have someone to help you with the analysis. And all of these things, they are actually quite competitive fields. And in order for us to have a meaningful contribution to these fields, it is important to be good at data generation, be good at AI, and be good at collaboration. And one of the things in Armenia that data generation itself, let's start from there. Like genomic data generation requires infrastructure and requires trained personnel who has experience working with huge amounts of data, with big recruitment cohorts, sample collection, etc. Things are moving. For example, there is a newly established sequencing facility in the Institute of Molecular Biology. And however, like it could have started maybe 20 years ago. It has just started and people are just starting getting experience with all the patient recruitment and the studies to generate data. And I do believe that we have opportunities to gather interesting data for generative AI as well in Armenia, which we cannot at the moment maybe have access to because of the lack of human resources and of course because of a lack of funding. The other thing is AI. And we have, again, scientists in Armenia and a lot of efforts to push this field forward. For example, there's Yerevan N laboratory, and there are many other teams that try to work with AI, and all of them are hardly working for towards fundraising, towards writing their own projects, and trying to be on top of their own fields. And with all of us in this survival mode, trying to push our own projects forward, it is hard to find a place where we can, work on an interdisciplinary project where we kind of say, okay, let's try and collaborate on one interesting project with both of our expertise meet. Because we're trying hard to, get funds for our own projects and keep our focus, because if we lose the focus, then it will be even harder for us to move forward. And that basically the lack of critical mass of people is something that doesn't allow us to push this field forward in the pace that we need to. Because if we do something that doesn't require a lot of infrastructure, for example, we try to use reinforcement learning, we try to use our genomics expertise, we try to develop a agentic model that does bioinformatics. That's something that we can theoretically do, but that is something that will require from us to leave all our projects aside and do only that. And the reason is that there are only a handful of scientists capable of doing that, and you cannot just leave all the projects behind and start moving in that direction, especially considering the pace at which the world is moving in these fields. And if you're slow, then you're not there. So basically it is hard to compete.
Thank you, Lilit. Unfortunately, every time we discuss science in Armenia, we have this common denominator that critical mass is what's lacking, and it seems like we have the diagnosis, but what do you think should be the top sort of priorities over the next 5-10 years to overcome this problem? Obviously, we can't just continue, uh, talking about the problem. There should be, projects or I think the government should somehow be involved in triggering, um, have a national program, educational initiatives to get the younger generation interested in science, in computational biology, in genomics, in biostatistics. These are definitely the future of have a great potential in the future. So, what do you think should be the priorities in the next 5-10 years to solve this problem? And before you answer, I see your hands. Um, Please, let's continue. Let's give our panelists the chance um, to continue the discussion, and then I will open the floor in about 20 minutes, and you can ask your questions, and I'll also read the questions from the chat. So let me give them a chance to reply.
Well, to my opinion, I think we need to prioritize the national goals in terms of science, not general, but in terms of science, we need to have some priorities. I mean, and these priorities should be not just on the white paper, but priorities depending on the national interest and depending on where we are going for the next 15, 20 years. It's not about next 3, 5 years. Because if you look at the government documents, there are very, very good documents for planning the next 3, 4, 5 years, and that's it. And and even if you dive deeper into that kind of document, you see absolutely no link between the science and the national interest, development of economy, development of other fields. It's like separate island science where some people define some priorities. I have no idea based on what. Um, and as a result of that, of course, we have what we have right now. Uh, so Lilit talked about bioinformatics, some different AI groups, and so on. During her speech, I just checked the number of publications that we had last year on AI. It's like just 9 papers published in 2024. And we are just one position above Rwanda and one position below Senegal. So this is reality. I mean, when we are speaking about ambitious programs like building some, I don't know, big factories for AI, supercomputers, and so on. First of all, we should like sit down and look around what we have. I mean, we have 9 papers published per year on AI and a few more on computer science in general. So, and the reason why we have such a situation is because we don't have any goals and we don't have any priorities towards to that goals. And government, they didn't spend a lot of time and programs to develop some specific fields. I mean, we are speaking about AI in general, and we know that AI is something fast developing, at least in the last 5 or 10 years. But what we did as a country or as a government to develop data science or AI in the country, how many labs we have established in the last 10 years focusing on machine learning or AI. So maybe 1, 2. I don't know more. So, but even if we establish some laboratories, so the result you can see 9 papers per year, and this is what we have. So we need to develop human pipeline. We need to develop some specific fields where we may have some advantage in the next 10, 15 years. But the reality is that right now we don't have any advantage. So we are just one position above Senegal or Rwanda. This is where we are right now. And we need to be very realistic. If we want to have some better position in the future, some ambitious programs in the future, we need to develop human pipeline. We need to have many, many labs working in universities or outside of universities in different research groups, working on very, very interesting topics, doing a very high quality science. And this we can achieve if government um, just prioritize the fields and start to develop specific topics or specific fields in science. Alternatively, we will still have the situation what we have, just one position above Rwanda.
Thank you, Hovakim. Any other comments? If you could prioritize one investment for Armenia that would move the needle, just one gauge, what would it be in terms of developing AI infrastructure and genomics research? Hovakim mentioned, having goals, long-term goals, and investing in, human capital and establishing laboratories. Is there anything else?
Yeah, I think there should be basically priorities are important considering that the resources are not infinite. And given the small resources, you need to understand which are the priorities and distribute them in those focused areas. And this will give an opportunity to have bigger investments. And bigger investments are needed to bring new blood from abroad, basically, to Armenia. Sarkis, you mentioned about attracting young talent. And I think attracting young talent is much easier. And there are a lot of projects that move in that direction. And for example, in ABI, we right now have more than 20 young investigators that they are very happy, they're motivated, and there are a lot of students, a lot of talented students that are out there, and we can like work with them. And the young talent is no longer the problem. It was the problem maybe 5, 10 years ago, but not right now. Right now, the problem is the middle age. Unfortunately, there are not the programs are not well budgeted enough to be able to bring an established scientist, say over 40 years of age, with quite a lot of experience, to Armenia. And do it in a clustered manner where you also recruit a team with postdoctoral and PhD students. And basically, that is the issue because we cannot, we kind of seem to not have the resources to supervise these young talents enough so that they don't leave the country because we don't have enough senior scientists in the field. And though the government is doing a lot and a lot of private initiatives, they are doing a lot. Maybe that's a numbers game and also a focus game. So if you have bigger grants that are targeted on specific forms, modes of hiring and specific research directions, maybe that will kind of turn the needle in the right direction.
Seems like a race against time here. The field is advancing at the speed of light, and generating the human capital is a time-consuming process. I have actually a question from Sevan Yasayan in the chat, very relevant question to what we're discussing right now. So I would like to bring this up. He says, based on limited understanding of the training challenges facing aspiring scientists in Armenia, it seems that workshops in computational biology, machine learning, and core biological and chemical principles could provide valuable opportunities for students. In addition to ABI's summer program, are there other workshops available for Armenian high school and university students? And is there a significant demand for these types of programs among students aged 16-25?
Since Evan already mentioned the summer school, I think that one is enough from our side. We don't target younger people, unfortunately.
The workshops or boot camps that is oriented to train people on AI, bioinformatics, chemoinformatics, or whatever, they're very useful. I mean, definitely there is need to organize such workshops, like I don't know, one month or 2 month workshops that can train people on bioinformatics or chemoinformatics. But it's like some very light solution of the situation. It's not permanent. So the solution should be to integrate new education programs into university programs. So if you have university, if you have faculty of chemistry, faculty of pharmacy, medical pharmacy, and so on, but you don't have chemoinformatics in that faculty or program, it's really very, very big mistake and lack. So even if you organize 1 month workshop, it will not fill the gap that you have. You can organize, you can train like 10 or 20 people and then pick up 1 2 good youngsters from that training group, and they will join you and you can work with them. But this is not solution. This is maybe solution for certain company or ABI or some institute to solve the current problems that they have with the lack of good talents. But in general, it will not solve the problem that we have in the country. We don't have enough human capacity. And if we want to develop that human capacity, we should go back to universities and try to modernize their programs, try to change their programs and make them according to current needs that we have, not only in the country, but globally. So because if you don't have updated programs, if you don't have state of the art programs in science and in education, so at the end you will have the situation that we have, and you will look around to see who is going to organize some new workshops or boot camps that you can join and get some good knowledge. But sometimes people are not lucky; they don't have Lilit, for example, who is organizing new workshop. And if they don't have Lilit, what will be with that people? They will not have any chance to get new knowledge on bioinformatics or some other important fields. So we need to solve the problem from the beginning, not try to find some light solutions for the current situation. This is what I think about human capacity in general.
This naturally brings me to the next question, and we here at ARPA Institute are heavily engaged, and for 33 years, like Dr. Panosyan mentioned, we're trying to help advance science, research, development, R&D in Armenia. But oftentimes we come up with initiatives. But I would like to pose the question as, what do you think and how do you think diaspora and scientists in diaspora, and we have experts, all over the world. How can we contribute? How can we support? Can we be involved and engaged to develop this, to help you uh, uh, in this projects, to advance the educational programs, to engage, for example, workshops. We can utilize the technology, online virtual platforms to do workshops, and support the ABI summer school and other institutions as well. What do you think specifically we can do? We can come up with hundreds and thousands of ideas, but it's important to hear from you specifically for the genome project, the genomic studies, for the virology, for the grapevine, initiatives. What is your take? We have a large audience here, mostly experts in different fields, and some are also in genomics field. So this is your moment. What would you like us to do?
So, if I can say so, since I am working in quite specific field, and a huge part of research and the data generated by my group was done, I would say, only thanks to the cooperation, long-lasting cooperation with European institutions. I'm always looking for partners because this is, as I told, very specific field, and we really need knowledge. We need a new knowledge. We want to go ahead to the new knowledge. and to meet some new challenges. And I'm looking around if there are some Armenians working in different well-known institution, let's say key player in that field. So unfortunately, I was not able to find someone in Davis, for example. I have a lot of colleagues, but not Armenian. So if there is someone who can share the information, so because as you understand, we have a quite multitask projects. We are not only focused on conservation, on biodiversity. We really need people who are working also in wine biochemistry, for example, in wine chemistry. So we really need a good specialist in that field also, because we are working on wine grapes. We're discovering new one. We need to understand if there is any potential. Do we need to conserve these wines, or we need to shift to other ones? So, and in that terms, yeah, the specialists working in different topics, as I said, especially wine biochemistry, we would be very happy to cooperate them because we have also a lot of ideas in that terms. And as I told, sometimes the challenges we are faced, it's not only related to the budget. Sometimes we have a budget, but we lack human resources. There is no people who would like to come to work in this very specific field, because we are not only doing a lot of wet lab work, we are going a lot of to the vineyards, and this is I guess the only problem, that the young generation, they don't want to go on to work out of the labs. So this is the only challenge I met in my group, to enlarge the group, I mean. We are looking really to start operation because we want to start a microvinification also. And there is no specialist in Armenia who will start to work on this very specific project.
Got it. Well, thanks for that, Christina. We have a question from Susan Jerian. She says, for grape wine project, what about collaborations with universities in California that have strong agriculture departments and substantial Armenian diaspora population? Have you tried to find and reach out here in California?
With California, no, we have so many different cooperative projects with Germany, Italy, France. We just started new cooperation with very specific topics, again, related to the phylloxera, genomics and the genetics with Bogu University. We just on the process to sign a contract, but with California, no, I have a good contact and cooperation with Davis University to Dario Cantu, but not with California. But we are very open and welcome everyone who want to cooperate with us. So,
right. At this point, we are hitting a 1 hour and 23 minutes mark. So I'm gonna move forward and open the floor for questions from the audience. And I have Mr. Ashur Petrosyan that have been patiently waiting for the last
Okay, but
20 minutes.
let me, if I can just add one thing. Just let me go back to your previous question about what diaspora can do for supporting Armenian science and education in general. If you know the history of Armenia in the last 100 years, especially the Soviet time, so probably you remember that in 1920s, many brilliant Armenians came from different places to Soviet Armenia, many scientists among them, many brilliant experts among them, and they started to create the country from zero. So you know that. So I think I always value the collaborations between Armenians from Armenia and diaspora. this is something that we really very much appreciate. But I think the best way to support something in Armenia is to come, live, and work in Armenia. I mean, it doesn't matter, it's about education, science, specific industry fields, or whatever, culture, art. So if you want to support your country, the best way is to live in the country that you have. So if we want to support science, of course, what ARPA did or what ARPA does right now, it's really very, very important. But probably the best way is to live in Armenia and do science in Armenia, do education in Armenia. And this is the only way how you can transform the environment. I mean, you can bring new values from, I don't know, United States, from Argentina, from China, from Russia, and change the values in Armenia. Alternatively, remotely, you cannot have impact as much as you want. The only way to change, to make changes in environment is to live in that environment and bring new values to that environment. So this is what I hope to see in the future.
That's absolutely true. I totally agree. Mr. Ashod?
Yes, can you hear me?
Sure. Go
Yeah,
ahead.
I'll be quick. I have an academic question, but I wanted to thank all of our scientists who work so hard, and you are truly heroes of our people. And hang tight and tough, and build the country. Don't be disappointed. It's you just have to develop creative collaborations. For example, one collaboration that we are involved with is this $100 million facility that makes potent oncology drugs in California. It's very hard to find such entities. And so how big is Armenia? Armenia is 3 million people, doesn't have that many people. So the company that we are working with makes oncology drugs for brain cancer and pancreatic cancer, the two killers. So Armenia will end up getting all of this medication for free. So it's like you don't have to worry too much about money. You don't have to build a $50 million facility in Armenia to do this, which we are thinking, I'm thinking, that will generate tremendous amount of revenue. So collaboration would be good. We have two gene therapy products that we are trying to work. If Armenia has any gene therapy products, we can collaborate. Just let me know. And the question that I had is what defines Armenian is the DNA, at least one aspect. It's been constant, I guess, for 5,000 years, maybe 10,000 years. So you know better what genetic diseases or traits are there among Armenians, if there is any, that needs to be eliminated and essentially a gene therapy cure developed. That's something that we would be interested in to know and work with Dr. Arsen of the Institute of Molecular Biology and all of you. Is there such a thing, or am I just mistaken?
I think Arsen is in this chat. I have noticed him. Maybe he can take it.
He is hiding. So basically, in this gene therapy, when Arsen comes, he can take over.
I think where I can maybe respond. So of course the obvious answer to this question is Yerevanian disease, the familiar Mediterranean fever, and that is the most logical target for Armenia. The challenge with this gene is that it's a long gene, and there are many different mutations and at different locations that can lead to the disease development. And genetic engineering in the classical sense, like base editing, will not work here. So you would need whole gene replacement therapies. And yeah, so I believe that it is something that actually our science team is trying to work on with some groundwork toward that. But of course, whole gene therapy is a field in its early in maturation stage. Yeah, so I'm not a gene therapist, although we had a recent project in collaboration with this institute at Harvard, where we developed a tool for uh, development of adeno-associated viral vectors as vectors for gene therapy, and we just developed a bioinformatics tool to help the scientists develop better vectors with better tissue tropisms, etc. Yeah, but I believe if Arsen Kuti would respond better to any gene therapy applications that are happening in IMB.
Yes, basically there are three types of viruses, the adenoviruses, and then so you need to share some knowledge as far as how big the virus is, how big the gene is. Usually for bigger genes you use lentivirus, and then we have all the capabilities to grow and purify and all of that. So I don't want to make this a technical session, but if this is something of interest, I'm in contact with Dr. Arsen. So if you contact him and then we can call it. But remember though, you don't need, like these folks want to clone express is $10 million. Well, where does Armenia bring $10 million? I mean, if they're going to do their own cloto, which is kidney malfunction gene, we just throw this one in there and basically happens. So we need to collaborate with diaspora, as Sargis was saying, and also the United States, which is the leader on gene therapy, biotechnology, and all of that stuff, and the places in California. And I'm in Orange County, California, so we're not that far. Thank you.
Thanks.
Great, thank you. If Arsen is available later on, if you want to comment, we'll bring him on. Moving on, we have already a bunch of questions in the chat, so I would like to take the time now to address those. The first question is from Gary Antinian. He says the Center for Medical Genetics in Yerevan performs genetic testing for targeted cancer therapy. We all know that the use of genomics and AI technology has challenges of implementation. Those challenges are diagnostic, consistency, limited government support, limited resources of personnel and lab facilities, and high cost. Do you have any ideas or plans how to make these technologies accessible to the public?
Okay, I can speak about
accessible to
that we have. So it doesn't depend on the public data. It doesn't depend on diagnostic consistency or laws or personal lab facilities and whatever. So recently we have developed the software with user-friendly interface. So which means that any researcher from Armenia or outside, from anywhere, if he or she gets access to the software, she can use it and design small molecules using the technologies and algorithms that we have behind the software developed by our team. So it's like about democratizing AI, because this is a big problem, and it relates not only to problems of data limitation or some other problems mentioned in the question, but it simply relates to skills that people have in the labs. If you look at classical labs from the United States to Europe and China and Japan, most of the labs that are working on drug discovery, they don't have data scientists, they don't have AI experts, they don't have even computational chemists or biologists in the lab. They're more classical wet labs that are performing different screenings using classical methods based on cell models or animal models. So these people need to have new AI approaches or softwares integrated into their research pipeline. So this is what we are trying to do. We are creating user-friendly interface-based software that people can use by just simple clicking the buttons and doing all this generative stuff, designing small molecules or optimizing small molecules without having any data science or AI knowledge. So this is, I think, something very important. So and this is our like contribution to giving access to AI tools that we have developed. So regarding other issues that we have, maybe Lilit or Kristine can answer better. But in our case, so we don't have these problems. The problem is that many research labs, they don't have experts, they don't have AI specialists and data scientists that can use new state-of-the-art approaches and integrate them successfully into their pipelines. Therefore, I think the best way is to try to develop some software that can have this user-friendly interface, helping people to use it without any limitations.
Thank you. If you want to add anything, Lilit or Christina. Okay. Our next question.
Yeah,
Absolutely. Go ahead.
I think I don't have anything more to add because I agree with Hovakim that the field should develop, can develop using the data that is available from elsewhere. But I didn't really understand the question, what should be made accessible to public? But yeah, if I didn't understand the question, I cannot contribute.
It was regarding the genomic
data.
genomic data at the Center of Medical Genetics in Yerevan.
I think it was the question was about technologies because the genomic data cannot be accessible to public since it is personal data that the Medical Genetics Center is handling. Yeah, so.
Right. Obviously, there are limitations in terms of making the genetic data accessible to public, but I guess for scientists it should relatively be accessible for research purposes. But nevertheless, that was the gist of the question. Our next question here is from Paul Nargisian. He says, regarding building research capacity and collaboration, what are the most promising ways to build sustainable genomics and AI research capacity within Armenia's universities? How can Armenian institutions join global collaborations on equal footing rather than as a data providers only? Followed by another question which says, how can Armenian University create ethical and culturally informed frameworks for managing genetics data? Are there national efforts to develop Armenia-specific genomic ethics or data shaping guidelines? I can repeat it at any point if this is too long of a question.
Yeah, I think I can take this. And basically, there are maybe three different layers. One is, again, human resources, which we have extensively discussed during this panel. Education is the other one, and the infrastructure is the third one. So infrastructure is basically, if you do have the capacity to generate this data, where do you store it, and how do you make sure that the data is used and shared in safe manner, and also like all the ethical guidelines are preserved. Right now, the laws in the Republic of Armenia that govern how the genetic data is shared are basically combined with biological data, like sensitive biological data in one pocket. Usually in different countries, genomic data is handled differently because differently than the clinical data, because your genomic data is not only your genomic data. You share your genomic data with all the people in your family and that share the same DNA material. So, for example, in Armenia, if you say, I'm okay with my personal data to be published somewhere, and you are the owner of your own data. In case of genomics, actually you cannot really tell it. But unfortunately, we do not make any distinctions at that level, only maybe under GDPR. We can make those distinctions. And this is not a very highly regulated field. The infrastructure is basically the servers where you store this data with all the required technical technical considerations to make this storage safe and anonymized. And basically, right now we have a server that we acquired during the first 3 years of our operations at the ABI, and that is shared with IMB, that IMB also contributed to. So basically, this is a shared infrastructure where we keep the data safely and we deal with the data in an anonymized fashion. And once we are ready for the research to go into publication, then the data is uploaded to public repositories. And yeah, this is something that we have to work with. One of the ways to do that was actually to join the international networks of bioinformatics data sharing. And one of those networks that are closest to us is called Elixir. It's the network of European bioinformatics institutes, where each country serves as a node. And one or a few institutes in each country are contributing to that node. And this network, basically, if you're a part of this network, then you as a node are responsible for making sure that all the European standards for dealing with genomic data are implemented in your own country. And this in turn gives you an opportunity to apply to all the grants that are a part of this Elixir network and also get some funding from there. And in order for us to be a part of this network, the government should make the first move. Basically, the government should tell that they're interested in joining the network, and there is a fee, annual fee, that the government pays, but it's not very much because they also take into consideration the GDP, etc. And yeah, this is something I think that we have to do in order to be a part of this network. And the next step would be to have a national server where we actually make sure that the data doesn't leave the country, but it is shared in a federal manner. So like all the metadata is shared, and if anyone from abroad wants to get access to the generalized data, then they can do that for research purposes. Yeah, but that's something that we're working on, unfortunately, privately at the moment. So there's not a government-led initiative to organize all of this. And the rest, how we develop genomics, how we develop BI in terms of human resources, I think we have already extensively discussed.
Great. It would be interesting to see government also involved in this conversations of developing private infrastructure for safely keeping the genomics data inside the country. And obviously, well, there are, this cloud platforms that, we can collaborate, like you mentioned. But I think it's important to have our own database that is safe. And well, moving forward, in the interest of time, I'll read out one or two more questions, and then we'll conclude this session. I'll give you 1 or 2 minutes to each of you to kind of summarize and give your final words. So our next question is from Shant Messergian. He says that with the recent investments from NVIDIA, Cisco, and recently also Firebird, right? How is the government and/or science community establishing opportunities to recuperate the benefits and thus reinvest into the next generation of scientists to resolve the brain drain towards non-science industries that was discussed earlier? And this is something we haven't touched yet.
Yeah, I think that's a part of a problem that there is a lot of infrastructural investment in Armenia without uh, equivalent investment into the brains, into the human resources. And of course, being close to an AI hardware is an opportunity for us and an advantage that we should be able to use. But I think there is a lack of understanding of how important it is, considering this investment, to start working on the human capital that can actually utilize all of this. So thinking about, okay, which projects could make us leverage all the resources in the best way? And what kind of expertise, what kind of experienced researchers do we need in order to undertake these projects? And what do we need to do in order to train these researchers or to bring them from abroad, etc.? Unfortunately, there is a mismatch between these investments and the investments that we see that are happening in order to bring the human capital on board.
Excellent. Thank you. Thank you, Lilit. We have a question from Mr. Ashot Petrosyan again. Am I right?
Yes, I'll be very quick. Dr. Arsen answered me in the text, so we are all set. We don't need any gene therapy for the Mediterranean fever. But be very proud and be very strong on the talents that we have in Armenia, and don't worry about the Armenians that are in diaspora. When the money is right, they will be on the first plane back to Armenia. So I was able to sell a $50 million pharmaceutical production facility to the Chinese that are in the United States based on Armenian talent. They said, "What does Armenia have to offer to us?" I said, "We have tremendous amount of education and knowledge, from the past, from the present. So I wouldn't worry about the resources. I know it's an issue, but once you start the project, believe me, the talents will also come. But these are applied projects. I mean, making brain cancer drugs, making pancreatic cancer drugs. It will generate tremendous amount of revenue for Armenia. So we are working on that project. We will see what happens. Thank you.
Thank you. So in the last 10 minutes of today's panel, I would like to give each of you a chance to, based on today's discussion, to summarize and recap and of the issues that we've discussed, and what are the most important priorities that you think can move the field forward. Take a minute or two just to recap and say your final words, please. Let's start with Kristine.
Yeah, thank you very much, Sargis. In my case, in my opinion, what I will need for my long-term projects for the next, let's say, 10, 15 years, the only thing I need is a human capital and involvement of the government. I hope that once the dialogue between science and governmental bodies must take place. And I hope that once I will get order from governmental bodies to do some project, and I will not, I don't need to apply to different funds to continue my projects, because when you are working with bioinformatics data, with genomics data, that's another question. When you are working with plants, you need the budget, you need the funds constantly. So you don't need each 2-3 years to apply and to hope that once your project will be funded, that you will be able to finish your project. So what I need, that's a human capital and a dialogue with government.
Thank you. Hovakim.
Thank you.
I think. Oh,
Go ahead.
Oh,
Ladies first.
you go ahead. That's okay.
do you know that was an order? So basically, I think this whole panel discussion was a kind of a pessimistic one, so I wanted to give it a bit of a positive note. I usually think in a way that you need to be prepared so that when the opportunity comes, you are able to grasp it at full potential. And I think this is what we are doing right now. And I think the preparations are successful in a sense that if we take individual examples, then we see a lot of initiatives in physics, in machine learning, in genomics, and other areas that I'm not familiar with, that middle-aged scientists like us, they come to Armenia actually from abroad, or they stay in Armenia after they are training abroad, and they contribute by developing talent and culture around them. And the example of the Armenian Bioinformatics Institute that I can bring, basically we started with a handful of scientists under 5 people, and in 4 years we trained a lot of people, and some are still with us. Like we right now have 20 plus people, the research staff at ABI, and a lot of them are actually studying in universities abroad, like in Stanford, Oxford, etc. And this is a development of human capital that is much, much faster than what we had before. And this is the groundwork. Basically, when the opportunity comes, when somehow, I don't know, $300,000 drop from the sky, and we're able to have a call for cluster hiring to build another research team, then we will say that we already do have the environment that will make the work of this team possible. So what we're doing right now are preparing the groundwork, and I think we are and not to say excelling, but we're quite good at it, because we are doing research that is connected to the world. It is research that is interesting to scientists around the globe, and we are developing talent, and there are like local environments that will make this shift possible. So hopefully then later on the opportunities will come, and we'll be able to make use of them.
So,
Thank you for the positive note, Lilit. Thank you.
So let me just tell a very short story. I told you about our position, just one position about Rwanda. So in the beginning of 2000s in Rwanda, their president, Paul Kagame, so a very contradictory person. Until now, he's the president. So he constructed the fiber cable network in the country. It's like 25 years ago. In a country where there were only like 300 users of internet, so 25 years ago. And people at that time thought that this is some corruption program because of some companies that just paid for that program. But the fact that after 10 years of constructing the fiber cable network in Rwanda, hundreds of IT companies came to Rwanda and started to form the ecosystem of IT in Rwanda. So in a small country. So this is about long-term vision. So if you have a long-term vision, if you have a country ruler's statements that have long-term visions that plan the country for the next 15, 20, 25 years, and plan it based on the data, plan it based on the analytic research, not just based on some hype, like fancy things like academicity and something like that. So in that case, of course, you can have achievements at the end. In contrast, if you do something just because of hype, because AI is popular, and you are saying that we are investing in AI because AI is everywhere, then in that case, at the end, you will not have any success. So, and the same thing is for scientists. I mean, they need to be very long-term visionaries. So they need to make high-quality science. There was a question in the chat about what Armenian scientists can do to have equal collaboration with foreign researchers or foreign scientists. For equal collaboration, you should have high quality science. I mean, at least in your mind level, you are not limited. You can do high quality planning in your mind, start to think on the questions that are really very interesting, and try to find collaborators that are also interested in that kind of questions as well, and start to make collaborations and do high quality science independently what you have at the moment. I started my lab in 2016 without having anything, just like two empty rooms. And then my German collaborators, they sent a lot of equipment, used second-hand equipment, and we started to use that equipment to do our first experiments. And then gradually we grow and get more equipment. And right now we have state-of-the-art lab allowing us to do all the antiviral research that people can do anywhere. So it's about how you set your priorities and what kind of interesting questions you are trying to explore. And if you have really very interesting, high quality questions, then of course you will find researchers, collaborators that will be interested to join you in that journey. So this is my answer to you to your question.
Sarkis, if I may make a couple of comments. I heard too much about government involvement. I think in any country like the United States, for example, the government doesn't do much to advance science, technology, and other areas. It's the initiatives coming from the individuals. You need the government by itself cannot do much. You have to have the right people with the right initiatives, with the right marketing. You're talking about, needing a lot of students. How come Lilit was able to find so many new students? And for example, Christina, you were not able to find. You have to analyze the situation and see what can you do to bring in students to your own research facility, train them, work with them. So my assessment is that you also need marketing of your work, and make them know, make everybody or the new students know about what you're doing, bring them in, show them what you're doing, and attract their attention. Marketing is very important. We've been talking about marketing in the clean room, for example. We're telling them that this is for the past 3 or 4 years. We're telling them that a clean room is a system. You need a marketing person, you need a technical person, you need an advanced scientist, and all these different positions need to be filled in order for a clean room to work. The same thing is for any research area. You need to do the marketing task. You need to let people know what you're doing and teach them what are the benefits of what you're doing, and therefore they will come to you. And the other thing is, as Hovakim just mentioned, and he answered to his own question actually, that if you have important questions that you're working on, people will come to you. And of course, they have to hear about your important questions in order to come to you. They have to know what you're doing. So that's another thing that I wanted to mention. So again, this is what I wanted to say. Sarkis, take the lead.
Absolutely. I think marketing and being out there in a social media nowadays is critically important for spreading and sharing, scientific information. And myself being a wet lab scientist, I came to realize at one point that really going out there and showcasing your work not only at conferences and meetings and etc., but also the having a website or going to discussions and showcasing the work to get the younger generation to learn about it. The information is now not in textbooks, but it's rather in on the screens, on the platforms, on the social platforms. So I totally agree that we really need to make effort to advertise, research, science, R&D, development, all of this in Armenia is critically important, and I'm sure this is being done. Like Dr. Panelists already mentioned, Lilit has done an incredible job recruiting talent in the genomics and data sciences. So I think this can be expanded and developed in other fields as well. And I don't want us to close this in a pessimistic note, right? Like Lilit mentioned, and I think we just need to build momentum. Yes, we need critical mass. That's important. We need collaborations and we need funding. All of these are important. But I think we have the potential. We have the talent, even though that talent is not, in Armenia predominantly. But we can come together in a form of panel discussions like this, and we can continue discussing this and raising the most important critical questions to the level of where we can come to a common ground, find collaborations, just like we witnessed one developing right during the panel discussion here. So these are all important. So I would like to just go ahead and extend our gratitude again on behalf of our to our panelists again for this exciting and important discussion. Again, Dr. Kristine Margaryan, Dr. Lilit Nersisyan, and Dr. Hovakim Zakaryan. I would like to also thank you, all the attendees, and all the interesting questions you've raised. I think this engaging platform um, and caring attitude towards science in Armenia is what is going to make the uh, uh, is going to help Armenia to come out of this, I don't want to say crisis, but a difficult and challenging situation in our history, and hopefully we will able to succeed together. Thank you all.
Thank you, Sargis, and thank you, panelists. This was great. Please donate if you haven't donated already, because we need money to do more in Armenia. And the second thing I wanted to mention is that if there was any other government that was doing this much, as much as this government is doing, I think we would have been in much better situation. So you need to help the government. The government needs to help you. It's a it goes both ways. It's not only the government that can change the country, but it's the people more than the government, I believe. Thank you very much. And we have a panel discussion on the Kemalist era and Armenia on December 27th. So please join us then. Thank you again, and hope to see you next time.
Thank you.
Thank you.
Thank you.